You know, for decades, the name of the game in fertility preservation has pretty much been more is better. But what if that's the wrong game entirely? Today, we're diving into a really huge shift in how we think about fertility, moving beyond just a simple head count to really understand what makes an oocyte viable. So let's dig into how this whole idea is being flipped on its head. And this is the question that's right at the heart of everything we're talking about today. For patients, for clinicians, the goal has so often been to retrieve the maximum number of eggs, or oocytes. I mean, the logic seems simple, right? More eggs equals more chances. But as it turns out, new science is telling us we might need to rethink that fundamental assumption. And this slide, it just lays out the paradigm shift perfectly. We're moving away from that old metric, oocyte count, which is a pure focus on quantity. And we're stepping into this new frontier of oocyte competence. And this is all about quality. It's not just about how many eggs you have, it's about their deep, intrinsic ability to create a healthy embryo. So the really crucial question becomes, how do you measure something you can't even see?
Okay, so here's our game plan for this explainer. First up, we'll look at the traditional goal of just counting eggs. Then we're gonna expose what we're calling the quantity blind spot, what that simple number totally misses. After that, we'll see how AI is stepping in to assess true potential. And finally, we'll take a look at what this new era in fertility is really all about.
All right, let's dive into section 1, the goal, counting eggs. We need to set the stage here and understand the traditional approach that has really dominated fertility preservation for years. So to get inside this traditional mindset, let's look at a recent clinical trial that our source material breaks down. Researchers were comparing different trigger strategies, and that's the final hormone shot that's given to mature the eggs right before they're collected. And their main goal, as you can see right here, was really simple: get the most oocytes possible.
Now, what's really fascinating here is what this table shows, and maybe more importantly, what it doesn't. The study found there was no big difference in the number of oocytes between the two trigger strategies for certain low-risk patients. Now, that's an important finding for sure. It confirms that one type of trigger is safe and gets you the numbers. But look closely. The key finding is all about quantity. The study answered its question, but the question itself was limited to just a head count.
And that leads us perfectly into section 2, the quantity blind spot. This is where we're going to unpack why that simple count tells such an incomplete story. And boom, here it is. This is the pivot point. This quote, from the analysis of the very study we just talked about, hits the core problem. The investigators themselves acknowledged it. Even the best studies working under this old paradigm have this critical limitation. They can count, but they can't actually assess the quality. So when we say quality, what do we really mean? Well, the scientific term is oocyte competence. And as you can see, it's the oocyte's inherent built-in ability to do its job, to fertilize successfully, to develop into a healthy embryo, and ultimately to result in a live birth. It is the biological potential that's locked inside that cell. And here's why this is so tricky to measure. Competence is determined by all these factors that are completely invisible to the naked eye. We're talking about things like cytoplasmic maturation, which is the overall readiness of the cell's internal machinery, or the distribution of mitochondria. Those are the cell's tiny power plants that fuel all of early development. You just can't see this crucial cellular machinery by looking through a standard microscope. Okay, so we've got a pretty big problem. The most important factors for success are invisible. This brings us right to section 3, AI assessing true potential. This is where technology is giving us a non-invasive window into that oocyte quality. This is where artificial intelligence is becoming a total game changer. Instead of a subjective human assessment, machine learning models can analyze a standard image of the oocyte. They can see patterns and features the human eye just can't pick up, giving us standardized, objective, and repeatable metrics. It basically takes the guesswork right out of the equation. So how does it actually work? Well, it's a pretty clear 4-step process. First, a standard image of the oocyte is captured. Second, the AI model gets to work, analyzing key structures like the cytoplasm and the polar body. Third, based on all that detailed analysis, an algorithm generates a competence score. And fourth, this score lets clinicians prioritize the oocytes, ranking them by their predicted potential. So why is this so critical? Here's a hard number for you. Typically, only about 80% of oocytes actually survive the freezing and warming process. That means, on average, 1 out of every 5 are lost. Now imagine if you could predict which ones were most likely to survive before you even freeze them. That's a huge real-world problem that AI is poised to help solve. And its potential goes way beyond just survival. More and more evidence shows that these AI models have incredible predictive power. They can help predict the likelihood of an oocyte reaching that crucial blastocyst stage of development. They can predict that post-thaw survival rate we just talked about, and they can even suggest the best orientation for microinjection during fertilization. These are seriously powerful and practical tools. All of this leads us right to our final section, a new era in fertility. So let's look at how these data-driven insights are set to create much better outcomes for patients. So what does this actually mean for patients in the clinic? Well, the impact could be massive. It means more informed counseling, where we can align patient expectations with the actual biological reality of their eggs. It allows for a truly personalized strategy, helping a patient decide if another cycle is actually needed. And by prioritizing the best oocytes, it could lead to improved efficiency and optimized resources, which could reduce the emotional and financial burden of it all.
And it's really important to get that AI isn't working in a vacuum here. The big idea is to combine the AI's visual analysis with other advanced data, like metabolomics and transcriptomic signatures. When you put it all together, you create this truly multidimensional assessment of quality, giving us a far more complete picture of potential than any single metric ever could. Honestly, it's the definition of precision medicine.
So here's the bottom line. This changes how we even do the science. In the past, like we saw with that Dono trial, studies focused on oocyte quantity as their main goal. But in the future, clinical trials are going to be designed completely differently. They'll use these AI-derived competence scores as a key endpoint. We'll finally be able to ask not just how many eggs, but which strategy produces the best eggs. This quote from the source material just perfectly sums up where all of this is heading. This isn't just a new tool, it's a fundamental reorientation for the entire field of assisted reproduction. The goal is shifting from a focus on number to a much more meaningful emphasis on competence. And that brings us to our final thought. For so long, fertility has been a numbers game, a game of chance. But what if the future isn't about collecting more lottery tickets? What if it's about identifying the winning ticket right from the start? This technology suggests the future is about finding the best chance. And the question for all of us is AI the key that's finally going to unlock it?